Hello, everyone. My name is Andrea Ruiz-Jones, and I am currently in my last year of undergraduate studies here at UC San Diego, where I study cognitive and behavioral neuroscience. I am a research assistant in Dr. Brad Wojtek's laboratory in the Department of Cognitive Science, working under the mentorship of PhD candidate Michael Preston. My research focuses on the neural mechanisms involved in cognitive processes such as vision and attention. By studying the electrophysiological components of neural activity, we can leverage quantifiable data to better understand these cognitive processes. Today, I'm going to talk to you about my research on analyzing stimulus-evoked changes in aperiodic electrophysiological activity in the macaque visual cortex. We will first talk about the background and our research question. Then I will tell you about the aims we had for the project, and finally, we will conclude with our current limitations and future directions based on our results. So, day by day, we receive thousands of sensory stimuli that are encoded in various regions of our brain. An example of this is the taste of a sandwich, encoded by the taste buds in our tongue, which then that information travels to multiple cortical areas in the brain. If you pay a little attention, you might be able to hear the footsteps of people outside this room, or even outside this building. And if we pay even more attention to these processes that sometimes we dismiss as background noise, we may become aware of the sensation of the clothes we're wearing against our skin. These sensory changes in our environment are reflected as stimulus-evoked responses in electroencephalograms and local field potentials, commonly referred to as LFPs. While EEGs are non-invasive and LFPs are, these measures of brain activity allow us to obtain information from event-related potentials, such as these sensory changes, with great temporal precision. But are these stimulus-evoked electrical responses that we observed functionally relevant in their entirety? Or perhaps certain components of these brain oscillations don't show a response when presented with a sensory stimulus? The analysis of electrophysiological brain signals has revealed the existence of synchronous and asynchronous oscillations in the brain. Those synchronous periodic oscillations, popularly known as beta waves, alpha waves, etc., have long been studied in cognitive processes such as vision and attention. However, recent human intracranial EEG studies have highlighted the presence of dynamic shifts in non-oscillatory aperiodic activity in response to visual stimulation. Previous studies have also demonstrated a link between aperiodic components and neural firing rate, along with the balance between inhibition and cellular excitation, using local field potential simulations. In recent years, there has been an increasing interest in understanding the functional and physiological significance of aperiodic activity, linking it to biological mechanisms such as aging and human vision. Despite this, the biophysical mechanisms underlying aperiodic activity have not been fully characterized. Non-human primate models allow us to investigate the cellular basis of local field potential activity. However, anatomical differences in visual cortices between humans and non-human primate models could cause differences in stimulus-evoked visual processing. Therefore, our first objective is to define whether the primate visual cortex exhibits changes in aperiodic activity related to visual stimuli. Additionally, we are interested in characterizing the biophysical mechanisms of stimulus-evoked aperiodic changes. Our hypothesis is that there will be changes in aperiodic activity related to visual stimuli, specifically due to the possible connection between aperiodic activity and the nature and strength of the LFP power spectrum, reflecting excitatory drive. To investigate these hypotheses, we utilize a publicly available dataset containing local field potential recordings from 2 macaques implanted with 1,024 electrodes in the primary visual cortex B1 and the supplementary visual area B4 in a resting state. 16 arrays, each containing 64 electrodes, were surgically implanted, and after recovery, the subjects were presented with a signal-to-noise ratio task. The task consisted of fixating on a blank screen for 400 milliseconds. Then, a checkerboard visual stimulus was presented for another 400 milliseconds, followed by a blank screen once again. As is standard practice in the field, the dataset was externally pre-processed with a low-pass filter, cutting out any signals above 150 Hz. Then, the data was downsampled, and the experimenters applied a bandstop filter to account for the electrical hum and its harmonics. Simultaneously, the multi-unit activity was also pre-processed to reduce noise and enhance frequency that are commonly believed to be related with brain activity. Both LFPs and multi-unit activity are extracellularly recorded signals from a local network of neurons, but LFPs are believed to be generated by membrane currents of the neurons in the local neighborhood of the recording electrode with low frequencies of less than 500 Hz, while multi-unit activity, or MUA, represents the spiking of local neurons at a high frequency of above 1000 Hz. We then used the multi-taper method on the LFP data only to compute a power spectrum that would help us parameterize both periodic and aperiodic components. This method multiplies the time series data by different tapers and averages the power spectral density results to reduce noise. We are interested in computing the power spectral density because it represents a metric of how a signal's power is distributed across different frequencies. This is how the power spectrum looks like after averaging. To do so, a mathematical approach called Fourier analysis is used to decompose the signal into its different frequency components. From there, the power spectrum is calculated using the squared magnitude from the fast Fourier transform, just like this. And that's how we end up with our power spectrum. The decreasing power with increasing frequency creates a particular 1 over f shape and prominent peaks which reflect neural oscillations. This metric takes our periodic activity, reflecting it as peaks in the spectrogram, such as the one we can see around 10 Hz, and the aperiodic component is reflected in multiple components, such as the offset and exponent of our 1 over f fit. Now, our lab has developed tools accessible to all public to parameterize neural oscillations and aperiodic activity from the power spectrum in a semi-automatic manner. This way, we can parameterize periodic activity as the oscillatory peaks presented in our power spectrum and the different components that represent aperiodic activity, such as the offset and exponent. The exponent parameter reflects the pattern of aperiodic power across frequencies. For this particular instance, we applied a spectrum, which outputs the aperiodic parameters we were interested in, the offset and exponent. But we also get periodic components represented as the peaks found in the analysis. Now, these results are only a representation of a small time window of one electrode of one trial out of the 1,024 of our entire multi-trial session. Hence, to calculate the overall stimulus-evoked changes in aperiodic activity, we needed to calculate the power spectrum for each time window, electrode, and trial in a way that would look similar to this graph, where the blue rectangle represents our time window, and we can see how the power spectrum changes across time. After plotting our data as such, we were able to visualize the power at different frequencies for our entire experiment, and saw that effectively, at time zero, when the visual stimulus is presented, there is an overall increase in power that, for some frequencies, decays after a few hundred milliseconds. Our spec-param time result results for aperiodic parameters and the goodness of fit of our model both also showcase a change in activity after stimulus presentation for both of our subjects. To further analyze these results, we examined the power spectrum differences prior and after stimulus presentation. We observed an increase in power in all frequencies on average for all electrode arrays. This was seen both for monkey A and for monkey L. We computed the difference in offset and exponent for each electrode after stimulus presentation to characterize the changes in aperiodic activity and found that most electrodes across B1 and B4 exhibited some response to the visual stimulus. This implies that visual processes may not only be encoded in oscillatory neural activity, but also in aperiodic activity. Inspired by our results, we decided to tackle our second aim, to characterize the biophysical mechanisms of stimulus-evoked aperiodic shifts. For this, we decided to investigate the higher frequency spiking present in the multi-unit activity recordings and found different degrees of spiking elevation between our subjects. Monkey L depicts an overall increase in neuronal firing while Monkey A also depicts changes, but not as prominent when looking at the average change in multi-unit activity. Nonetheless, when analyzing only the post-stimulus windows, we can see increased firing in both subjects. Therefore, our next approach was to compare the post-stimulus spiking activity that we had previously analyzed with the aperiodic offset and exponent, and we saw that these components are inversely correlated in the post-stimulus window, meaning that higher spiking activity will induce a flattening in the power spectrum represented by lower offset and exponent values. To take a closer look into the relationship between these components, we investigated the spiking activity and the aperiodic offset and exponent in a time-resolved manner. Here, we plotted the z-score difference for each of these values across time and saw that all of them displayed an increase right after stimulus onset. But the aperiodic components decayed around the 200 millisecond mark. Since our previous results were an average representation of the post-stimulus window, that later decay could explain the inverse correlation we see between the aperiodic components and spiking activity. But further investigation needs to be done in order to determine the validity of this hypothesis. Finally, I'd like to discuss some of the limitations that we found during our research and future directions of the study. In summary, we found that the macaque local field potentials in the primary visual cortex B1 and supplementary visual area B4 do exhibit stimulus-evoked changes in the aperiodic activity. The encoding of this visual stimulus is associated with broad increases in the aperiodic offset, and these changes may be linked to underlying spiking activity, yet further analysis still needs to be done. Understanding how stimulus-evoked responses are functionally encoded in non-human primates and comparing these processes to what we currently know from non-invasive human models can open doors to using non-human primates as models to study cognition and understanding the neural mechanisms behind visual encoding, since these models offer a more feasible access to study invasive methods in healthy populations compared to human studies. Additionally, current human studies are limited in patient populations and placement of electrodes determined by medical condition. One limitation we found during this study was that pre-processing filter applied by the experimenters who collected the data might be affecting the results of our analysis, given that specific frequencies are being suppressed because of these filters. Therefore, as part of our future directions, we are planning on refining our analysis by looking into the goodness of fit metric fluctuations in our spec param results and using the raw data to only apply a low-pass filter, since spec param already accounts for the electrical hum that caused the experimenters to apply the bandstop filter during pre-processing. Additionally, we also want to characterize the interaction between aperiodic shifts with oscillations and event-related potentials, given that increased firing could help us better understand the relationship between synchronic and asynchronic neural activity. We also believe that exploring eye signal data and pupil dilatation across sessions would be interesting, as it might serve as a possible biomarker for attention and excitability, which could be linked to aperiodic shifts. Thank you so much to everyone for this incredible ride. I want to thank particularly my lab, the Wojtek lab, my PI, Bradley Wojtek, and my mentor, Michael Preston, for the continuous support that I've received throughout this entire project. Thank you.